Hi everyone, my name is Andrew Byrne. I'm a process chemist here at APC. My co-presenter Iselt Mossop. Iselt is a process engineer here at APC and we're both based out of Dublin, Ireland. Today we're going to talk to you about continuous processing and PAT for chemical development and iterative design. To give a high-level overview of the content we will cover today, in section 1 I will discuss configuration screening and flow optimization. Here we will touch off quick screening for appropriate reactor selection, screening within these novel kinetic areas that are accessed through flow processing, and a how-to with regards to setting up unsupervised screening with PAT to accelerate your process development. In section 2, Iselt will discuss PAT and flow processes and what to do when you get that data, the importance of integrated PAT for data intensification, and a how-to with regards to creating inexpensive flow rigs for proof of concept lab campaigns, and the implementation of feedback control in these flow rigs utilizing your PAT. Presented on the current slide is an overview of APC and Our Partners. APC is a CRO, or Contract Research Organisation, based in Dublin, Ireland. APC employs over 140 scientists and engineers working across a range of modalities from small molecule to large molecule. We partner with large biopharmaceutical and pharmaceutical companies around the world to accelerate the delivery of their medicines to the market through process development, and ultimately accelerating the delivery of that medicine to the patient. Through a synergy of chemistry, biology, and engineering, we offer value by enabling rapid process feasibility, process definition, development, and characterization, so our partners can accelerate the development process. The advantages to implementing flow chemistry and continuous processing configurations are well documented. These advantages include increased safety, increased quality, such as yield and purity. These increased qualities are often achieved by reaching the novel experimental window enabled by flow chemistry, the integration of unit operations, increased productivity, and all while reducing that all-important footprint. APC employs an iterative process design strategy to enable process intensification via the conversion of batch processes to flow. Our teams of scientists and engineers utilize this framework to move from concept onwards towards strategic planning and design, to proof of concept, and eventually onwards to the next generation. An informed conversion strategy and design allows the team to move swiftly to proof of concept work with integrated PAT and offline analytics on our range of in-house screening platforms with both custom and off-the-shelf solutions. Of course, depending on the phase of the molecule in development, phase-appropriate work packages can be added or subtracted. Following our successful proof of concept, the next steps would include the integration of unit ops, feedback control, machine learning, design of experiments, or the development of hybrid solutions, depending on the needs of our partner. Over the next section, configuration screening, I'm going to take you through some of the aforementioned steps we typically employ while taking that leap from batch processing to continuous processing. Data-rich experimentation is key to our approach here at APC, in batch and in flow, but more importantly, in our case, for batch informing flow. We must understand the reaction characteristics, the critical quality attributes, and this will help us inform the continuous reactor design and configuration selection. I suppose APC chemists are a little bit different. We're trained in the use of PAT-enabled systems, which allows us to accelerate that data mining process and characterization of reactions. The targeted use of PAT combined with our in-house expertise of chemistry engineering affords data-rich experimentation, which really means you get a higher yield of information per experiment. The application of FTIR, Raman, and FPRM affords live reaction data, allowing us to track catalytic reactions, heterogeneous or homogeneous reactions, and easily characterize these. We can screen from 1 mil to 25 mil scale, and this is really attractive because for early phase projects, we often have a low material availability, and with these 1 to 25 mil volume reactions, we really don't need a lot of material to get a high information yield. It is this information that we then feed into the next stage, which is configuration screening. Following our batch process technical assessment, we now know how our chemistry performs. Is it homogeneous or heterogeneous? Does it use a catalyst? Is it mixing limited? How quickly does it reach completion? We have most likely trialed FTIR, Raman, UV or LC as online methods of characterizing and following this reaction. All these factors in the data collected influence reactor design and selection, whether that be PFR, packed bed, CSTR, or a combination. Configuration screening can then take place. This can take place on a range of platforms, like our off-the-shelf Vaportec or custom platform rigs as seen above. As previously mentioned, often the scale of these reactor systems can be scaled to meet material restrictions. On completion of the small-scale reaction screening, it will be often clear which configurations are favourable for flow and proof of concept. And to be honest, in my experience, it is often very clear which will work and which will not. On the current slide, we can see a ranking system for each of the individual configurations screened for an alkylation reaction. And we can see which configurations give acceptable purity and acceptable yield, but it will often be very clear, as in this case, which configurations give a superior yield or purity. In the above case, it can be seen that a dual PFR system is favourable with an inline quench of the reaction intermediate with either a packed bed or a pre-dissolved quench. And this was tracked by an in situ FTIR probe and verified using HPLC data. So why is PAT so important to our approach here in APC, and especially to this iterative process design for flow? Let's look at the classical experimentation method presented on the above slide. Without PAT, the scientist must be present, and there may be gaps or delays in obtaining results. And often, particularly in overnight periods, we can see that there is no experiment conducted or data collected. Now moving forward to a PAT-enabled scientist, process analytical technology combined with chemistry and engineering allows unattended, inline, real-time analytics, allowing chemists and engineers to make informed decisions faster. Combining PAT with our in-house development expertise allows us to rapidly screen configurations and conditions in flow chemistry. A synergy of offline and inline analytics is key to this success and allows us to automate the screening of reaction conditions. This slide details the integration of PAT into one of our screening platforms used for the feasibility and proof of concept studies here at APC. In this case, it's a Vaportec using an FTIR flow cell, but also this could be a UV flow cell or the other selected PAT that would be identified during the batch screening process. Typically, inline HPLC, UV-Vis or FTIR can be used post-reaction or in between reactors in series in our flow chemistry rig. Combining this technology with some coding and automation allows rapid parameter investigation, greatly reducing development time and costs, often allowing automated experimentation overnight. In the example presented, we used a jacket temperature of 150 degrees Celsius to access the novel experimental window above the boiling point of the reaction solvent for this particular case study. In this alkylation, we harness PAT, namely FTIR, to monitor a reaction intermediate in this alkylation. Using IC Quant, we were able to build a univariate quant model to identify and quantify a specific reaction intermediate based on peak height. Using feedback control, we were able to optimize the concentration of the product in the output liquors and minimize the remaining starting material. Through automation, we were able to do this throughout the lab day unsupervised, but also to carry out some experimentation overnight. This PAT-enabled system was later combined with our online HPLC skid. The presented graph presents an overlay of the obtained online HPLC data compared to our inline FTIR. The concentration measurement via FTIR at 3 different conditions and residence times overlays comparably with our measured HPLC data. It is this synergy of PAT and traditional analytics that adds value and speed to our development workflow. To give a really high-level summary of the impact of moving from batch to flow for the aforementioned alkylation, I've just put a very quick summary together on the current slide. In moving from batch to flow, we increased our yield by 7% and our purity by 9%. This was all achievable by reaching that novel operating window at 120 degrees, enabled by increased pressure from our flow system. The increased temperature reduced our reaction period or residence time from 24 hours to 30 minutes, drastically increasing our productivity. But there was also a surprise in that the reaction was more efficient at these higher temperatures, reducing the requirements for higher levels of input reagents. So we were able to reduce the amount of feed B by 1.2 equivalents. So by converting from batch to flow, we effectively increased our yield, increased our purity, but as a bonus, we were able to reduce the cost required for reagents and, of course, that all-important footprint. I'm now going to hand over to Iseult, who's going to take a deeper dive on PAT and flow.
Thank you, Andrew. My name is Iseult Mossop, and I am a chemical engineer working with APC as well. So I'm just going to switch gears a little bit now and talk about a bit more of the nuts and bolts of creating a simple flow rig for a few minutes. It's easy to get daunted with the idea of creating a small proof of concept lab model to investigate a flow chemistry process. Most of the rigs that we see in pictures look extremely complicated and expensive. However, simple flow reactors for lab scale proof of concept work can actually be completed quite inexpensively with off-the-shelf components, particularly when investigating reactions at ambient temperature. So this is what we're going to talk about for the main bulk of this section of the talk. So take, for example, the rig in this picture. With simple peristaltic pumps, which I'll highlight now, balances and PFA tubing, we were able to create a simple flow rig, which performed an acylation reaction. This rig actually also included some online monitoring and was also built out to include feedback control, which was driven by the Mettler-Toledo universal control box, which is hidden back here. I'll speak a bit more about the feedback control loop now, but this photo kind of gives you an idea of how inexpensively and simply these rigs can be built to demonstrate just proof of concept. As I mentioned, all components in this rig were sourced from off-the-shelf suppliers, and there's nothing bespoke or expensive included in this rig. It really is just pumps, balances, a bit of tubing, and a few T-mixers. And by combining all of these elements together, we were able to create a proof of concept demonstration of a flow acylation reaction. One challenge that we did face with this acylation process was that we had to overcome a highly mixing sensitive environment. So the process was extremely mixing sensitive due to the fact that feed A was organic based and feed B and C were aqueous based. Therefore, when poor mixing was presented, a significant impact on the yield was observed. A simple solution could be implemented to overcome the laminar mixing in the lab scale model. One such way of overcoming this is to include a small static mixer like the one in the photo here. These static mixers are available from a number of off-the-shelf vendors, and they're quite inexpensive. Due to the scale that's often incurred with these sort of lab models, there's severe material restrictions apply. So therefore, we can't just replicate the kind of mixing environments and the flow rates that we would have on scale. So therefore, turbulent mixing is often not achievable. By including one of these static mixers, we can replicate the kind of mixing environments that we would see on scale without jeopardizing the material, the limited material that we might have. So if you can see from on the left-hand side, there's a CFD image, which I'll just highlight. So this CFD image shows you that when you have two opposing streams entering, which don't mix very well, you can see here in the red and the blue, the streams are extremely separate, which leads to the poor yield. So once they hit this static mixer, you can see that there's a disturbance in the flow, and it's creating a higher or a better mixing environment for the two streams. And as they move through this static mixer and down the tubular reactor, you can see that towards the end we have a fully mixed stream, which means that the mixing is obviously, the two streams are mixing a lot better. So in this example that we're talking about right now, this actually incurred a yield increase from 60% to 90%. So really the mixing environment in this case was highly important. And by using this simple trick, we were able to replicate the mixing environment that we will see on scale. So it's just important to note here that sometimes these issues that we have at our lab scale won't actually occur on larger scale, but we do need to address them so we don't try to scale in an inappropriate way. So we're just going to move on to a different example now. As Andrew mentioned earlier, one of the major advantages of flow processing is accessing those novel kinetic region. So this is generally done by increasing the pressure to suppress the boiling point of the solution. To monitor these processes, therefore, it's necessary that high pressure sampling is implemented. In this example, in this photo here, we have a two-step continuous process, which includes an extraction unit here over on the left and a crystallization unit on the right. Now, to monitor this process, we actually included an additional vessel to this process, which is a high-pressure Easy Max in the middle here. We included this because we wanted to implement an Easy Max probe or an Easy Sampler probe to monitor the supersaturation of the extracted solution before it hits the cooling crystallization. It's really important that we include this online monitoring because otherwise it'd be very difficult to gain the information required to complete a focused and informed optimization of the process. If we were operating in a traditional batch unit operation of this nature, offline sampling is next to impossible due to the high pressure nature of this system. Generally what would happen is that the extraction would occur and it would immediately then be either moved into a separate vessel or the crystallization would happen in the same vessel, therefore impossible to get that information. Therefore, not only will this flow process increase the throughput, it will also add data intensification and understanding to the process. So for semi-batch or stirred tank-based flow reactors, like the one in the last slide, accessing different pressure zones can be easily implemented by adding nitrogen to the headspace. However, when we move into tubular reactors, this can become slightly more problematic. However, we can implement a simple trick which can be used to access different pressure zones in a tubular reactor. An expensive investment like a dynamic back pressure regulator can be avoided by using a peristaltic pump, as these pumps act like a one-way valve while releasing pressure at a stable rate. So this pump can be placed at the end of the tubular reactor prior to the product collection point to control the reactor at a higher temperature. For example, if the combined flow rate of the exiting the reactor is 2 mils per minute, the pump would be set to 1 mil per minute, operating in the same direction as the flow, to create a controlled and stable pressure buildup in the reactor. You just need to be mindful that all other components are pressure rated, or you might have a bit of a mess on your hands. So how do we tie all of this together, from reaction flow screening to building a simple flow rig and incorporating PAT? The aim of converting to a flow process is that the throughput can be scaled out as well as up, which means that the process can simply be run for a longer period of time. As every process is dependent on the quality of the upstream unit operations or the feed material that you're supplying it with, they must be designed to account for changes or disturbances and react to them with minimal time lost. The potential incurred by operating in a bad zone in one unit operation, causing a knock-on effect in downstream unit operations, can be catastrophic. As all steps are connected in continuous processes, this is why feedback control is so important for these unit operations. Although feedback control loops are important for batch operations, they're inherently less risk involved, as all of these steps are not telescoped together, and the material is generally separated and tested prior to feeding the next step. The first step in building a feedback control loop is identifying a suitable way of quantifying the parameter of interest, the parameter which the feedback control loop will use to control the process. In this case, an in situ IOR was identified to measure the product, the starting material, and the main impurity. This is the next iteration of the project that Andrew mentioned previously in this talk. So the chemometric model was built to quantify these species, as the spectra for the starting material and product were very similar. Initially, to build out this feedback control loop, a universal control box was used to link the pumps and the balances to iControl, where a simple feedback control loop was created through the data link tool in Excel. We used basic logic statements such as, if X is greater than Y, then increase the flow rates by Z. Unfortunately, this initial approach, however successful it was, could only be used to control one feed stream, and we wanted to control 2 out of 3 feeds active here. So we had to build out a little more sophistication facilitated by moving to MATLAB. So by moving to MATLAB, the pumps were directly controlled by this software now. We had to decouple them from the iControl software. So as you can see from the flow diagram on the left-hand side, the data that fed into iControl really was just the IOR data and also the balances. These were exported then to Excel via the data link tool, and all of this information was then grabbed by MATLAB to feed into the algorithm where the actual feedback control loop was iterated. One of the major advantages of decoupling the pumps and the balances was that although MATLAB was changing the internal setting of the flow rate, which was determined by the algorithm for the feedback control loop, we actually had a live check of whether this internal set point on the pump was actually was being initiated correctly, and this was done by having the balance data feeding into iControl. So as I mentioned, the major reason for moving to MATLAB was that we were able to implement dual parameter control, and we also managed to implement an unsupervised startup. This meant that once we hit start on the software, the pumps were able to start up and begin to find the optimum space of operation for the desired conversion. A target of 80% conversion was specified within the algorithm, and this is represented by this high conversion limit here in the yellow line. This is guided by the IOR data that we see coming in the grey line here. So this represents how much product is actually in our solution stream exiting the tubular reactor. So as we can see from the overall plot here on the right-hand side, the final control system was able to achieve operating within that high conversion limit. We also see a disturbance towards the end of the experiment, where the conversion dropped below this 80% limit. The feedback control system quickly reacts to correct this. Finally, on this slide, I thought I'd just circle back to a photo of the rig that we're actually talking about with this feedback control loop. So as you can see, it's completely housed within a standard double hood. Again, it was a proof of concept demonstration, so we didn't want to build out very expensive capabilities when we weren't even sure if this was going to be a viable option for our client going forward. Secondly then, I'd also just like to highlight how sensitive this feedback control loop was. So if you can see on the plot on the right-hand side over here, we can see that when we had force disturbances in our product stream, each feed stream took an independent reaction to the force disturbance. The feedback control loop will constantly make adjustments to keep the target above 80% conversion, and this is whether you put in a different downstream product or whether there's a disturbance due to temperature or pH. It's important to note that these systems can be introduced at lab scale to showcase the value that could be implemented during manufacturing. We showed here that you can get a highly intensified process not only from throughput but also information. The information that we got from this process meant that each unit operation could be fully understood, and that it could make its own autonomous decisions on keeping the process on track. So that kind of concludes just a quick snapshot of what I'm going to talk about in terms of how to build out these simple controlled feedback loops and flow